Welcome. This video introduces nDNA, why it's needed, how it's computed, and what it shows. The pillar of neural genomics. We are starting to trust AI systems with real decisions, such as admissions, hiring, loans, medicine, and even policy drafts. To use them responsibly, we need a way to compare, audit, and govern their behavior over time. To certify models before deployment, monitor updates, and catch drift before it causes harm. Counting parameters or staring at attention maps doesn't answer those needs. Think of a model like a symphony. You could count instruments and pages of score, but what really matters is the performance, the way themes move, tempo breathes, and sound flows. We take the same approach with AI. We listen to the forward pass as the flow of meaning through layers. We contain that artificial intelligence is not merely an engineering construct. It is a digital, semantic organism with emerging artificial cognition, sculpted by data, objectives, and inductive priors. As genetics once enabled the life sciences to transcend taxonomy and uncover mechanism, we now require a similar epistemic leap in AI. We propose nDNA as that leap, a diagnostic grammar that exposes the hidden anatomy of understanding within machine cognition. nDNA is more than a metaphor. It is an emergent, high-dimensional scaffold of internal cognition. It is a latent topography through which behavior is realized. It introduces a rigorous diagnostic framework for probing the geometry of learning, capturing latent transformations that conventional benchmarks and surface evaluations fail to reveal. We read AI foundation models as semantic fluid dynamics, meaning is transported through layers like fluid through a shaped conduit. Along that journey, it bends, spends effort, and is pushed by internal currents. nDNA is the physics-grade telemetry of this motion. A geometry-first, coordinate-free fingerprint anchored to what the model actually digests and processes semantics. With it, we cross into biology, tracing lineages across pre-training, fine-tuning, alignment, pruning, distillation, and mergers, measuring inheritance between checkpoints, and spotting drift as traits shift. It enables us to describe phenotypes and infer genotypes, compare models, diagnose risks, and govern change over time. You probably know this, but before we proceed, a brief review of how a modern large language model operates.
Transformers typically also include a second type of operation known as a feed-forward neural network, and this gives the model extra capacity to store more patterns about language learned during training. All of this data repeatedly flows through many different iterations of these two fundamental operations, and as it does so, the hope is that each list of numbers is enriched to encode whatever information might be needed to make an accurate prediction of what word follows in the passage. At the end, one final function is performed on the last vector in this sequence, which now has had a chance to be influenced by all the other context from the input text, as well as everything the model learned during training, to produce a prediction of the next word. Again, the model's prediction looks like a probability for every possible next word. Although researchers design the framework for how each of these steps work, it's important to understand that the specific behavior is an emergent phenomenon based on how those hundreds of billions of parameters are tuned during training. This makes it incredibly challenging to determine why the model makes the exact predictions that it does.
Think of a transformer stack as a complex piping system through which a semantic liquid flows. On the left, the simulation shows how fluid moves. Sharp bends signal turning paths, narrow throats demand more effort to pass, and swirling eddies show competing currents. Now, the research question we ask, can we measure and characterize the semantic fluid dynamics within a transformer using coordinate-free reparametrization invariant observables? This is exactly what nDNA answers, by reading bends, effort, and push along the forward pass. Since not everyone comes from a physics or mechanics background, let's take a quick, plain dynamic tour of a few fluid dynamics ideas we'll use.
In laminar flow, the particle velocity is constant. The shape of the flow line is fixed. In general, laminar flow has a low speed. No energy lost. Satisfies Poiseuille's law. Meanwhile, in turbulent flow, the shape of the flow line is always changing at any time. The shape is uncertain, intersecting each other, some even just going around in circles. In turbulent flow, the speed of the particles is irregular. The shape of the flow line is always changing. In general, turbulent flows have high speeds. There is energy lost. Does not satisfy Poiseuille's law. Hopefully this tutorial can provide an idea of what laminar flow and turbulent flow are.
The nDNA score brings 3 insights into one readout. First, curvature shows where meaning sharply turns or warps. Second, effort captures the cognitive work the model pays to cross a belief boundary, how hard it is to change its mind. Third, push strength measures the prevailing current inside the model that steers responses, a signal of subtle bias or cultural drift. Together, these form a stable, behavior-type diagnostic you can compare across checkpoints and updates. See this figure carefully. On the top left, you see laminar flow. When a fluid is laminar, streamlines run in parallel with little mixing. In semantic terms, the model is refining a thought steadily. Low curvature, small and even effort from layer to layer, and the internal push agrees with the direction of travel. Think polishing a summary rather than changing topics. On the top right, you see spectral curvature, kappa. Now look at the wavy traces. A sharp local spike in kappa marks a turn in representation space, a pivot in meaning. Before and after the spike, the line is almost straight, so the spike isolates the moment the model reroutes. A topic jump, a shortcut, or a policy jolt. Curvature tells us where the path bends. Bottom left. Think of this plot as the model's effort curve. In a pipe, a narrow throat, or pumping uphill, requires more force to keep the same flow. Inside a transformer, a conceptual bottleneck does the same. Extra effort to transport semantic fluid. On the plot, the band thickens and lifts when effort rises. Short spikes are quick tolls. Broad humps are long climbs. These bursts often mean a stance change, resolving a contradiction or compressing many facts into one claim. On the bottom right, belief vector field, V. Think of these arrows as an internal wind. Sometimes a tailwind, sometimes a headwind. Longer arrows mean a stronger push. The direction shows where that push wants to take the answer. If the wind lines up with the path, the model moves smoothly and efficiently. If it blows across or against the path, the model hesitates, takes detours, or even circles. Those small loops are echo pockets, where it keeps revisiting the same thought. Put together, kappa marks the turns, L counts the effort to pass bottlenecks, and V shows the push and its alignment. This is the nDNA triad. We'll use these 3 signals to fingerprint behavior, compare checkpoints, and detect drift. Reading the flow, not just the plumbing. Now that we've covered the basics of fluid dynamics, let's translate those ideas into nDNA. Transformers are not real pipes, so we can't just drop fluid equations into a network. Instead, we keep the intuition, flow, bends, bottlenecks, and currents, and build behavior-type coordinate-free measurements. Here's how we make those adjustments. Think of nDNA as 3 instruments reading the flow. First, spectral curvature shows where the flow of meaning turns. Second, thermodynamic length measures how much effort the model pays to cross a representational boundary, how hard it is to change its mind. Third, belief gradients map the internal wind that orients the answer and pushes it forward. Together, they form a stable, behavior-tied fingerprint. Spectral curvature measures how sharply the latent manifold bends or twists across transformer layers. On the left, we see curvature values plotted by layer, highlighting segments where the model's internal trajectory sharply changes. The right 3D plot visualizes curvature across layers and data indices showing variation in semantic geometry depending on input data. This measure reveals how the model's internal representations evolve, indicating where significant shifts in conceptual understanding occur. Spectral curvature is thus a fundamental component of the nDNA score, helping quantify the geometric complexity of AI cognition. If this still feels abstract, let's use a simple cars and roads illustrations to build intuition for nDNA and semantic fluid dynamics. In this video, you'll see a pack of cars descending a hill. On straight, steep sections, gravity gives a push and they accelerate. On flatter or uphill bits, they lose speed. At sharp bends, they must steer and shed speed. Those turns are our stand-in for spectral curvature, the moments when a transformer's representation sharply changes direction as it moves through layers. Thermodynamic length quantifies the latent effort, measuring not just what the model knows, but how hard it works to adapt that knowledge across layers in response to epistemic pressures. Example, cultural fusion, alignment shifts. The left 3D plot shows how thermodynamic length varies across layers and data samples, highlighting zones of intense representational change. The right plot visualizes the thermodynamic length across layers, revealing regions where the model compresses or expands its internal representations, key points that reflect learning dynamics. This measure forms a crucial part of the nDNA score, illuminating how hard the model works internally to evolve its semantic understanding. Here the car is fighting the hill. Each meter costs more. Throttle goes up, traction wavers, speed dips, then recovers. This is the intuition behind thermodynamic length, an effort meter for the forward pass. When the model must restructure belief, resolve a contradiction, switch stands, or integrate long-range context, its effort rises just like this climb. Long, steady grades show sustained cost. Short, sharp rises are effort spikes at bottlenecks. In transformer terms, each layer is a stretch of road. Smooth layers let it roll. Bottleneck layers force a hard climb. We introduce a new lens in the form of the belief vector field, which captures how internal belief signals evolve across transformer layers. For example, the belief vector for justice may increase in magnitude across layers, indicating a stronger semantic emphasis, while the vector for disobedience might shift direction, revealing evolving contextual interpretation. As information flows through the layers, these belief vectors shift dynamically within the latent semantic space, revealing how the model's internal understanding and cultural priors guide its reasoning. This evolving field provides a powerful tool to interpret and audit AI cognition, helping us understand alignment, bias, and decision-making at a granular level. This iconic scene shows the car spinning while flying. Turning the path is curvature. Rolling the body is torsion. Torsion is a corkscrew twist along the route. In a transformer, high torsion means the representation isn't only changing direction, it's reframing, shifting stance, swapping point of view, or rotating which features matter. Low torsion means steady framing. High torsion is a spin in how the model carries meaning forward. In transformer terms, torsion is the representation rolling across layers. The model keeps moving forward, but the vector space that interpret tokens rotates. The Neural DNA score, or nDNA, is a cumulative measure that captures the latent geometry of a foundation model. It quantifies how the model adapts its internal cognitive beliefs by integrating three key components at each layer. First, curvature measures how twisted or sharply bent the latent manifold is, revealing semantic inflection points in the model's reasoning pathways. Second, length quantifies the latent work or effort the model expends as its internal representations evolve, indicating how hard it adapts its beliefs. Third, the belief vector norm captures the torsions of the model's belief signal in latent cognition, encoding how strongly cultural or contextual priors steer the model's internal space. Formally, the nDNA score is defined as the weighted sum across layers of the product of these three quantities: curvature, length, and belief vector norm. Though technically intricate, our proposed neural DNA, or nDNA, can be quickly understood as a unique fingerprint of AI cognition. It combines three core measures: how sharply the model's internal reasoning bends, curvature, how much effort it expends adapting its beliefs, thermodynamic length, and how strongly external, cultural, or alignment influences steer its thinking, belief vector norm. Together, these reveal the hidden geometry shaping a model's identity and evolution, offering a powerful new way to understand and guide artificial intelligence. For a deeper dive into the details, we suggest referring to the book. To understand how different and unique these semantic fingerprints are across large language models, we selected 15 distinct LLMs from diverse model families. This comparative analysis reveals their individual latent genomic signatures, highlighting variations in internal adaptation, reasoning dynamics, and cultural priors. This 3D plot visualizes the neural DNA signatures of models within the Llama family, comparing Llama 3 base, Llama 2 base, and Llama 3 instruct. Llama 3 base shows a higher thermodynamic length, meaning it works harder internally to refine its beliefs compared to Llama 2 base. This aligns with what we know. Llama 3 introduced architectural improvements and more training data, enabling deeper reasoning and more nuanced responses. Meanwhile, instruction-tuned versions like Llama 3 Instruct showed distinct patterns reflecting fine-tuning for specific tasks or conversational behavior. These differences in latent geometry give us a window into how model updates translate into changes in internal cognition. This 3D visualization compares the neural DNA signatures of Gemma base and Gemma instruct models. The higher thermodynamic length in Gemma instruct reflects increased internal adaptation effort during fine-tuning. Changes in spectral curvature reveal sharper, more focused semantic pathways, enabling better reasoning and safer outputs. Notably, the Mixtral expert model displays generally lower thermodynamic length than Mistral base, suggesting a more efficient internal adaptation process, possibly due to expert fine-tuning or mixture of experts architecture. Spectral curvature differences indicate subtle shifts in the model's semantic representation pathways, reflecting how specialized tuning alters the conceptual landscape. Compared to Quen base, the Quen instruct model shows increased spectral curvature and thermodynamic length, reflecting more nuanced internal transformations triggered by instruction tuning. This aligns with Quen's design goals to enhance contextual understanding and task generalization without exponential scaling. The heightened latent effort corresponds to the model's deeper internal refinement to handle complex instructions, supporting stronger alignment and robustness across diverse tasks. DeepSeek models showcase a profound relationship between their architecture and the latent geometric transformations that enable advanced reasoning. The higher spectral curvature seen in the chat variant reflects sharp bends in the latent manifold, marking key conceptual transitions essential for complex, multi-step reasoning. At the same time, the increased thermodynamic length measures the total effort the model expends navigating this semantic space, capturing how the model dynamically adapts its internal beliefs to interpret and reason over inputs. This integral of representational change embodies the model's epistemic depth and flexibility. Together, these geometric and energetic dynamics reveal that DeepSeek's reasoning is an emergent property of its internal semantic shape and flow. This perspective offers a powerful lens for understanding AI cognition beyond mere parameters or data. This 3D visualization compares the neural DNA signatures of a few models outside major families: Falcon, GPT-NeoX, Phi2, and TinyLLama. The spectral curvature and thermodynamic length across transformer layers reveal distinct latent geometries and internal cognitive dynamics for each model. Falcon, known for its high performance and open weight architecture, shows elevated thermodynamic length and spectral curvature, reflecting its capacity for complex semantic reasoning and adaptability across diverse tasks. GPT-NeoX, designed for large-scale distributed training and versatility, similarly exhibits rich latent dynamics, indicating deep internal effort to reconcile vast contextual information. On the other hand, Phi2, optimized for efficiency and smaller scale, shows more streamlined internal geometry, aligning with its design goal of delivering strong performance with lower computational overhead. TinyLlama, a lightweight variant aimed at edge deployment, naturally demonstrates simpler latent transformations and reduced adaptation effort, consistent with its compact architecture and efficiency focus. This comprehensive 3D plot displays distinctive neural DNA signatures for 15 language models spanning multiple families. It captures how spectral curvature, thermodynamic length, and belief vector field evolve across transformer layers, revealing unique latent geometries and internal cognitive dynamics. Each curve represents a model's signature, showing how semantic bending and latent adaptation effort vary by architecture, training data, and fine-tuning. One concept to keep in mind is the flatness of the cognitive genesis. The system can get flatter, stickier, and comprehensive only if it lives in flint state. This is why semantic bending and latent adaptation effort vary by architecture, training data, and fine-tuning. Notably, models like Mixtral, Llama 3, and DeepSeek exhibit elevated spectral curvature and thermodynamic length, signaling deeper internal reasoning and complex semantic trajectories. Conversely, lighter models such as Phi2 and TinyLlama occupy lower regions in latent effort and curvature, consistent with their efficiency and scale-focused design. Together, these signatures offer a geometric fingerprinting framework, enabling direct comparison of AI cognition, interpretability, and alignment potential across diverse LLM landscapes. At this juncture, you might have many questions. Why focus on these three measures? How task-dependent or language-dependent are these signatures? While we will address some of these questions shortly, a deeper exploration is available in the book. Briefly, DNA in biology is the universal code of life, governing development and traits across organisms. Yet, its expression is highly context-dependent. There, the same genome produces vastly different outcomes in different tissues, blood cells, eyelashes, or skin, adapting uniquely to each environment. For now, let us turn to the power of neural DNA, how it unveils entirely new perspectives on AI cognition, adaptability, and alignment. We invite you to explore how nDNA not only quantifies latent geometry, but also reveals profound insights into the evolving identity and reasoning dynamics of AI systems. Let us take you on a journey to glimpse the transformative potential of neural genomics. You may have noticed we're plotting only the last 10 layers of the transformer stack. A natural question is, why are we concentrating on the last 10 layers only? Most modern decoder-only LLMs have about 30 layers. Many papers try to map who does what by depth. This paper is my favorite one. The authors group LLM transformer layers into three regions. Think of the stack as a trip from raw words to a committed answer. Early, shared layers are the on-ramp. They build basic language features, tokens, grammar, simple relations. The flow is steady and mostly straight. Middle, transition layers are the interchange. The model starts routing meaning. It decides what's relevant, links distant parts of the context, and sketches a plan for the answer. This is where you see more turning and some resistance, choosing a route, reconciling conflicts, compressing details. Upper refinement layers are the decision zone. Here the model finalizes what to say and how to say it, following instructions, setting tone, applying safety rules, formatting, and committing to a claim. Small changes here create big changes at the output head. That's why nDNA focuses on the top 10 layers. This is where turns sharpen, effort peaks, and the internal push is clearest, making it the best place to fingerprint behavior, compare checkpoints, and catch drift. In short, early layers read, middle layers route, upper layers decide, and nDNA measures the flow right, where decisions are made. Think of the network as a production line. Early layers extract raw features. Middle layers compose them. The upper layers finalize the claim. That decision zone is where 1. The output head reads states, 2. Instruction tuning and alignment leave the strongest fingerprints, and 3. All three metrics agree. Turn semantic fluid into final meaning. Effort to cross boundaries and internal push become most concentrated and most predictive of behavior. Focusing nDNA on the last 10 layers gives a stable, behavior-tied fingerprint with far less compute. Our measurements also align with efficiency goals. The upper stack accounts for a disproportionate share of useful effort, so auditing there is both informative and frugal. When needed, new tasks, long-distance reasoning, or anomaly hunts, we can run full stem nDNA, but the top layer's view is the right default. Before we go further, a fair question, because nDNA is read from a model's behavior on a given input. Is nDNA task or corpus dependent? If so, in what sense can it be a unique fingerprint? Our answer has two parts. We find empirically what task changes actually do to the nDNA scaffold. We fix a baseline nDNA for identity, then measure delta nDNA for each task. Common sense, dialogue, QA, reasoning, and summarization. Tasks move the turns, change the effort, and reorient the internal push, but the core top layer signature stays. Identity is stable, state is quantified. The book provides a detailed account of our experiments and an in-depth discussion of the findings. Let us explain nDNA from biological DNA lens. In biology, genetic code is essentially the same whether sampled from skin, blood, hair follicles, or nails. What differs is how easy it is to read and the state marks each tissue carries. That's the nDNA analogy. The core fingerprint stays the same, while tasks and domains add state-specific variations that we quantify as delta nDNA. At the top is the idea of a shared genome. Every cell carries the same DNA, yet tissues act differently because regulators decide what gets expressed. On the left, DNA methylation tends to silence genes. Paired with neuron transcription factors, it opens the chromatin needed for neural programs and turns synaptic genes on. On the right, histone acetylation supports activation. With liver transcription factors, it promotes metabolic and detox programs, while neuron programs are kept compact and off. Same genome, different expression. That's how biology achieves clarity through complexity. This section draws a parallel between evolution and learning. In biology, DNA is shaped by selection pressures over many generations, while regulatory networks fine-tune which genes are expressed, so organisms develop robustly and adapt. In LLMs, pre-training plays a similar role. Stochastic gradient descent over massive corpora acts as the selection pressure, choosing latent priors and features that minimize expected loss across many situations. That objective is what you see in the equation, expected negative log likelihood, and the bracket below it reads, SGD as selection pressure. After pre-training, fine-tuning and alignment behave like epigenetic adjustments, modulating expression without replacing the underlying code. The takeaway, both DNA and nDNA emerge from selection plus regulation, one in nature, one in data, yielding stable codes that can be adapted to context. We conclude that what the genome is to life's functional unity, DNA is to a model's reasoning coherence. One shared code, selectively expressed and readable by its flow. The table below lines up the pieces. First row, the universal code. In biology, the genetic code maps three-letter codons to amino acids with built-in redundancy, so DNA sequence is error-tolerant. In LLMs, pre-training creates a shared latent code with useful symmetries. The reason a model can generalize across phrasing and style. That's the universal layer. Second row, context regulator. Biology turns genes on or off with transcription factors and epigenetic marks. Networks of regulators choose which program runs in a cell. In LLMs, the prompt and instructions select a latent path through layers. The internal route the representation follows, different prompts light up different routes. Third row, path geometry. Cells follow minimal effort routes across an epigenetic landscape when changing state. Models do something similar. They tend toward low-cost latent paths when moving from prompt to answer. nDNA reads this geometry via our three signals: curvature or turns, effort or thermodynamic length, and push or belief field. Fourth row, output mapping. Biology reads gene programs out into cell types, neuron, hepatocyte, and so on. Models read latent states out into task outputs at the head: answer selection, summary, dialogue turn. Final takeaway: same idea in both systems, a universal code plus context dependent regulation traveling along a shaped path to a specific output. nDNA gives us a rigorous way to measure that path in models. At this point, all my AI researcher colleagues and team members argue. Why these three signals, curvature, effort, and push? Couldn't other metrics do better? We tried several families. Here's what we found: norm-based pairs, activation norms, weight norms, singular values. These collapse under trivial rescaling and layer norm effects. They tell you how big things are, not how they bend or where they are pushed. Magnitude without structure, gradient-only diagnostics, Fisher diagonal, local logit gradients. They capture local sensitivity strain, but miss the shape of the path and the directional drift imposed by priors or alignment. You see stress, not the route or the wind. Entropy measures, activation or token probability entropy, helpful in addressing uncertainty and diversity, but primarily focused on the output side. They're weakly tied to the internal geometry that reveals inheritance or reorganization. Ad hoc pairings, example curvature plus activation variance, effort plus norm. These fragments tell the story. They don't jointly encode shape, cost, and direction in a single interpretable frame, and they yielded poorer separability across model families in our tests. This isn't the last word. There may be stronger composites ahead. It is an evolving science. Why not more metrics? Why not fewer? When we added extra dimensions, activation norms, entropy, variance, we mostly gained noise and lost interpretability. They didn't reveal new axes of latent variation or external forces. When we reduce to two signals, say curvature and effort, we lose the ability to localize external steering from alignment or cultural priors. nDNA combines three readings: curvature, how the path turns; effort, how much work it spends; and external steering, the push that orients it, and into one stable map, the model's unique fingerprint. For a more detailed discussion, please read the book. Historically, artificial intelligence has drawn its most profound insights from biology: the neuron. The brain's fundamental computational unit shapes modern AI architectures and learning. While this neurocentric view enables significant progress, it limits our ability to address critical issues like alignment faking, request denial, deception, and many more mystic traits of emerging artificial cognition. We must expand our lens beyond neurons and synapses to the genomic level, a framework that captures the latent and evolutionary dynamics of emerging and evolving artificial cognition. nDNA is a first step, not the last word. Yes, extraction methods will improve. I even sketch an nDNA v2 later, but the leap is already here. A coordinate-free, behavior-tied fingerprint that separates inheritance, mutation zones, and ideological drift where ad hoc metrics blur them. This marks the start of neural genomics, reading the semantic helix of artificial cognition, moving from surface scores to internal physics. With this fingerprint, we cross into biology, tracing lineages across pre-training, fine-tuning, alignment, pruning, distillation, and mergers, measuring inheritance between checkpoints. Now, let's walk through the evidence, how nDNA behaves across models and tasks, and the new routes it opens for comparison, audit, and governance. nDNA opens several new paradigms. We trace how cultural priors and language choice reshape the streamlines via cultural nDNA, CIVC, and multilingual analyses. We start observing model-level operations through the nDNA lens for alignment, quantization, pruning, model merging, collapse, and distillation. We monitor nDNA before and after such operations and measures what mutated. We ask evolution-style genomics diagnostics questions with a toolbox of measures: NHT, NGDI, NTDS, N-karyotyping, NDIV, NEPI, NCCL, each quantifying a facet of the semantic flow. distance, drift, bottlenecks, lineage, diversity, interactions, and cross-context consistency. We then revisit several NLP operations through the nDNA lens: machine translation, adversarial attack, multi-turn conversation, plus CAR and NEFOS. For each, we read how steering or stress reroutes the internal semantic flow, then design targeted edits to correct or amplify it. nDNA isn't just a microscope for mechanistic interpretation. It's a compass for shaping the evolution of artificial cognition. Please read the book and continue to the following pages and videos